Hello friends, welcome back to Medico Srinjoy. Today we'll study a very important chapter of the dental histology, which is the salivary gland. Without wasting time, let's start the today's lecture. What does these salivary glands? These are the exocrine compound and the tubular gland. So let's understand what does this exocrine means. Exo means outside and the crine means the bloodstream. So together this exocrine word means that the salivary glands release their secretion directly into the bloodstream, but in the ducts. So they does not release directly into the bloodstream, but they release their secretion into the ducts, and which carry secretion to the various parts of the body. Next, we'll understand what does this assignee means. That is assigner. What is this assignee means? Assignee are nothing but the basic functional and the terminal secretory units. So what are these assignee? Assignee are the basic functional or the terminal secretory unit of the salivary gland. This assignee is further can be divided of the two types, that is the serous assignee and the mucous assignee. So there are two types, serous assignee and the mucous assignee. Now, what does this serous assignee and the mucous assignee? Serous assignee are the compound alveolar gland, while the mucous assignee are the compound tubular gland. So let's study this serous assignee in detail. What are the serous assignee? Serous assignee are nothing but they are the compound alveolar gland, and they appear circular or rounded in shape. Further, they are smaller in size as compared to the mucous assignee, and their lumen is also small. Next, cellular assignee cells are lesser in number and pyramidal in shape. Also, the nucleus is round in shape and are placed basally 1/3 part of the cell. All these features are very important. Kindly remember this. Next, we have that the serous arsenic contains cyanogen granules which are stained by the H&E stain because they are eosinophilic in nature. Next, we will learn what are these mucous assignee. As we already discussed that the mucous assignee are the compound tubular glands. Also, they are oval or tubular in shape and they are larger and compared to the serous assignee. Further, the mucous assignees have the wider lumen and the cells are more in number and columnar in shape. Also, the nucleus is flattened and are placed in the basal plasma membrane of the cell. Apically, the cytoplasm is empty, so they doesn't take the H&E stain. Next, we will understand what are the various demilunes. Okay, what are demilunes? Demilunes are the half moons. So, what does demilune mean? These are the half moon shaped thing, and they are placed in the serous demilunes. Next, we will study what are the ductal system of the salivary gland. What are the ductal system of the salivary gland? The ductal system of the salivary gland consists of the secretory and the excretory portion. While the secretory portion modifies the saliva by exchange of ions, for example, the intercalated duct and the striated duct, and the excretory portion does not do anything, just they transfer the saliva. So they are like transporters. For example, the excretory portion is the excretory duct. So the saliva transports from the intratubular duct to the intertubular duct. Next, we will learn these three type of ducts in detail. First, we will start with the intercalated duct. What are these intercalated duct? These are lined by the single layer of low cuboidal epithelial cell with empty appearing cytoplasm. So they are having a single layer of the low cuboidal epithelium cells. Also, they consist of lysozymes and the lactopherins. These two things are secreted from this intercalated duct. These are the lysozymes and the lactopherins which are contributed into the saliva. Next, we will study the striated duct. These striated duct are the largest type of duct present. So these are the largest type of duct and they are lined by a single layer of tall columnar cells. So we need to remember that the intercalated duct are lined by the low cuboidal epithelial cell, while the striated ducts are lined by a single layer of tall columnar epithelium cells. Next, we will study that the striated duct have abundant large mitochondria and are radially oriented and located in the portion of the cytoplasm between the membrane enfolding. So this combination of the mitochondria and enfoldings gives the striations. So the striations of the striated duct are because of the membrane enfoldings and the mitochondria present there. Finally, the apical part of the plasma membrane show microvilli which project into the lumen. Here the isotonic saliva gets converted into the hypotonic saliva while passing through the striated duct. The reason is that the absorption of the sodium and calcium is higher than the secretion of the potassium and the bicarbonate. Also, water is not added, so the isotonic saliva gets converted to the hypotonic saliva. Also, the striated ducts provide the glycoprotein and calcarein to the saliva. So, these two substances, that is glycoprotein and calcarein, are contributed to the saliva by the striated duct. Next, we'll study the last part of the salivary duct, that is the excretory duct. This excretory duct consists of two layers. Okay, there are two layers. These are the mucosal epithelium layer, which are the pseudostratified columnar epithelial cells, and the outer connective layer containing adventitia. So the two layers are the mucosal epithelium layer and the connective tissue layer of adventitia. Also, brush border cells, dendritic cells, lymphocytes, macrophages are also present in the excretory duct. Excretory duct also releases a very important substance, that is mucin, in the saliva. So we need to remember that the intercalated duct secretes the lysozyme and lactoferrin. Striated duct releases the glycoprotein and calcarein, while the excretory duct releases the mucin in the saliva. Next, we will study the functions of the ductal system. First function is that the primary saliva, that is the isotonic saliva, gets converted into the secondary saliva or the hypotonic saliva. Also, the intercalated and the striated duct resolve some proteins, and the excretory duct modifies the tonicity of the saliva. Next, we need to understand a very important topic, that is what are the myoepithelial cell. This question is very frequently asked in the viva and the exam paper, so let's study this in detail. What are these myoepithelial cells? These are also called as the basket cells and are similar to the smooth muscle cells, but they are derived from epithelium cells. So that's why they are called the myoepithelial or the basket cells. These cells are located around the terminal secretory unit or the assignee of the ductal system. Also, these cells are stellate or spider-like with a flattened nucleus, and there are scantily perinuclear cytoplasm and long branching processes. These modified cells have the following functions. These are: they accelerate the initial outflow of the saliva from the assignee. They also reduce the luminal volume, contribute to the secretory pressure in the assignee, and also reduce the backflow of the saliva. And this also helps in the underlying parenchyma supporting. So there are mainly 5 functions. They accelerate the outflow of the saliva, they reduce the luminal volume, they contribute to the secretory pressure in the assignee, they reduce the back permeation of the saliva, and they also support the underlying parenchyma. So let's study the very important and a very frequently asked topic in this chapter, that is the classification of salivary gland. On the basis of the nature of secretion, salivary gland is of two types. These are serous gland, mucous gland, and the mixed gland. The serous glands are the parotid gland and the von Ebner's gland. The mucous gland are all minor salivary gland except the von Ebner's gland. And finally, the mixed gland is the submandibular gland and the sublingual gland. Also, on the basis of size, salivary gland is of minor salivary gland and the major salivary gland. The major salivary gland are the parotid gland, submandibular gland, and the sublingual gland, while the minor salivary glands are the labial glands, lingual glands, buccal glands, palatine glands, and the glossopalatine glands. Now let's study the major salivary gland in detail. The major salivary glands are of three types. These are the parotid gland. There are two parotid glands present. There are two submandibular gland, and there are two sublingual glands. So let's study the parotid gland at first. The parotid gland is the largest major salivary gland. So the parotid gland is the largest major salivary gland, and it has the following structures. The length is about 5.8 centimeter. The width is about the 3.4 centimeter, and the weight is about 14 to 28 grams. This weight is very important. Sometimes these are asked in the viva, that is, a parotid gland has a weight of about 14 to 28 milligram. So let's study the location of the parotid gland. The parotid gland is two parts. The superficial part lies in front of the external ears, and its deeper part lies behind the ramus of the mandible, fitting the retromandibular fossa. So this retromandibular fossa accumulate the parotid gland. Next, we'll study the ducts. As we know that all the salivary glands are having ducts. So, what are the ducts present in the parotid gland? The duct present in the parotid gland is called as the Stenson's duct. This is very important, that the parotid gland's duct is called as the Stenson's duct, and it opens in the cavity in the buccal mucosa opposite to the maxillary second molar. So it opens in the maxillary second molar. Also, it contributes to the 30% saliva production. Next, we'll move on to the submandibular gland. What are the submandibular gland? Their weight is about half of the parotid gland. Okay, so their weight is about 7 to 14 grams. That is half of the parotid gland. The location: these are located in the submandibular triangle behind and below the mylohyoid muscle, with a small extension lying above the mylohyoid line. Friends, if you like my video, then please subscribe the channel and hit the like button. So let's study the ducts present in the submandibular gland. The ducts present in the submandibular gland are the Wharton's duct. So the name is very important, the Wharton's duct, and it contributes to the maximum of the saliva production. That is 60% of the saliva are produced by the submandibular gland. Finally, we have the sublingual gland, which is the smallest major salivary gland, and they are located between the mucosa of the floor of the mouth and the mylohyoid muscle. There are two types of ducts. The main duct is the Bartholin duct, and also small ducts are also present, that are the duct of Rivinus. So there are two ducts, Bartholin duct and the duct of Rivinus. These sublingual gland only produce 5% of the saliva. So let's revise the ducts name once again. The parotid gland have the Stenson's duct. The submandibular gland have the Wharton's duct. The sublingual glands have two ducts, the Bartholin duct and the duct of Rivinus. Now let's study the minor salivary gland. Minor salivary glands are about 600 to 1000 in number. So they are very large in number, that is, they are about 600 to 1000 in number. The minor salivary glands are located beneath the epithelium in almost all parts of the oral cavity, except in two places, that is the anterior raphe of the hard palate and the anterior two-thirds of the dorsum of the tongue. Also, this minor salivary gland distinctly lacks the capsule. So they lack the distinct capsule. And the secretory units open via short duct directly into the mouth. So their ducts are not named separately. They are only named on the basis of their location. So they are named as the buccal, minor salivary gland, labial, lingual, palatine, and the glossopalatine. Okay, so these are named on the basis of the location. That is, if they are present in the buccal, they are called buccal. Similarly, the labial, lingual, palatine, and the glossopalatine. Friends, you can download the PDF note of the today's lecture in the description box. Next, we'll study the composition of saliva. This is a very important and a 10-marker question frequently asked in the university exam, that write the composition of saliva in details. So let's study the composition of saliva. The saliva has a pH of about 6.5 to 7.5 and a total volume of about 750 to 1000 ml per day. The saliva mainly consists of 99% water and the 1% the organic and the inorganic substance. This inorganic substance present in the saliva are mainly calcium, the phosphate ions, which protect the mineralized enamel from the dissolution, the fluoride ion, which promote remineralization, the bicarbonate ion, which act as a buffering agent, and the thiocyanate, which are the antibacterial in nature. The organic substance present in the saliva are mainly salivary amylase, which is a starch digesting enzyme, and they are majorly present about 50%. There are also salivary pellicle, they are 40 to 45%. There are mucins, and the lingual lipase, which are secreted from the von Ebner's gland, and the antibacterial enzymes are like the lysozyme, lactoferrins, antifungal substance like the histatins. Also, there are immunoglobulins like IgA, IgM, IgG are also present. Small amount of steroid hormones like cortisol, estrogen, testosterone are also present here. Some excreted products like the ammonia, urea, sugars like glucose are also present in the saliva. Next, we'll study the functions of the saliva. There are mainly 7 functions of the saliva. These are protection, digestion, mastication, lubrication, deglutition, taste, tissue repair, mineralization, and the cleaning of mouth and the excretion. First, the major function of the saliva is the protection. Protection, it protects the mouth by buffer system, which is contributed by the bicarbonate ion. The antibacterial agents like the lysozyme, lactoferrins, and histatins, the antioxidants like the uric acid, ascorbic acid, and the immunoglobulins like IgA. In digestion, the salivary amylase digests the starch, the lingual lipase digests the lipids, while the hydrolytic enzymes are also help in digestion. These saliva also help in the mastication, the lubrication, the deglutition. Also, the taste, saliva dissolves the food substance and it helps in the digestion. Also, the tissue repair are done by the saliva and mineralization. So friends, if you like the video, then please subscribe the channel and hit the like button. Share this video with your friends and share the knowledge. Next day we will study the very important and very crucial topic in dental histology, which is the oral mucous membrane. This oral mucous membrane is a very important topic because this topic are frequently asked in first prof exam and also are frequently asked in the third year, that is third prof exam. So friends, please like this channel and like the video.